All right. So this is a presentation that we gave for our Forest Service partners on October 18th for the Ecology and Botany meeting. My name is Emily Swindell, and I am the crew lead for the Native Plant Materials program. I've been trained in ecology, botany, geology, and GIS.
Yeah, and I'm Allie Clark. I'm the botany technician for the Native Plant Materials project this year, and I am an ecosystem science and sustainability major from CSU, and I just graduated this past spring. So just a quick overview about what we're going to be talking to you all about today. We're going to go over the program goals that we had this year, how we went about building the field program, the field work we conducted, the results that we gained, and then also the lessons that we've learned.
So to give you an overview of our program goals, essentially we were here this season to collect native seeds across Region 2 for Forest Service restoration efforts. And these efforts refer specifically to these two objectives. And this first one, the bipartisan infrastructure law objective, this means that one of the goals of this program was to ensure the integrated benefits for long-term soil stability, to maintain and improve wildlife habitat, and provide for resilient ecosystems through those efforts of collecting native seeds. This second objective, the burned area rehabilitation category 2 objective, is associated with the Office of Wildland Fire under the DOI, and it means that our program will help to ensure that appropriate native plant materials are available for use in post-fire efforts to repair and improve fire-damaged lands unlikely to recover naturally to a management-approved condition. Another goal of this program was to partner with Colorado Native Plant Society, and they were a crucial partner in this project because they are able to increase capacity. They can conduct education events through public outreach, and they're also able to advertise public collection events. So they're able to get a lot more hands out into the field with us to increase our collection weights. So they were very important in this project this season. And finally, we are here to help build a regional program. So that means we're here to help with data management, with spatial data analysis, also with monitoring assistance for in situ restoration projects in the future, in the years to come. And we're also here to support development of decision-making tools to help guide these collection efforts.
So on to how we built the field program. There was a lot of pre-season work that went on before Emily and I were onboarded. That was done by Jessica, Donna, Marty, and Maggie over the past several months. And they came up with a total of 18 species that we would be focusing on this year, 14 of which being grasses and 4 of them being forbs. And then we went into our training for the season, which included time spent in the herbarium, looking at pressed specimens, doing field walks with our forest partners and the specialists here at CNHP, going through ID books, and then also 1 day of Seeds of Success training. Some things that we built this year include a Survey 123 form and a field map. The Survey 123 form is based off of the Seeds of Success paper form with a few tweaks so that it's a little bit better for our specific program. And then the field map includes everything that we'd need for scouting. So sign-up points, collection polygons, move them in major road layers, the ranger districts, provisional seed zones, ecoregions, and also geology. And then we also went about creating a species reference guide, which just helps us go over some of the phenology, that location, some ID specifics for each species, just to help us get more familiar with those 18 species we'd be working with. And finally, there was also some logistical work that went on behind the scenes. So things such as gear acquisition, creating the safety forum, creating protocols for being in the field, as well as our scheduling.
We would start out by reaching out to our forest partners And this would start with an email from Donna or our supervisor, Jessica Smith. And they would send out some basic information about our program. So they would include the target species list that we just saw on the previous slide. They would send our authorization letter and also just an overview of the goals of this project. So what we were out there to do, what we weren't going to do. And then Allie and I would take over with logistical questions, such as who can be our safety contact on the National Forest while we're out there? Are there any places that we shouldn't be collecting from, like RNAs or research natural areas? Are there ground disturbance policies? Are there places that have already been revegetated or reseeded? What are grazing schedules? Where are shooting ranges? Things like that. And then we found that sending a post-field follow-up afterwards was really helpful for partners. This included what we collected, what we saw that wasn't ready to collect, species that we didn't see at all. And so this just gave an overview, a summation of our week or 2 weeks in the field. And we found that through this whole process as well, that flexibility was really key. We had partners that had the time and the knowledge of where to take us and to be out there in the field. Other times we had partners that were so busy with their other projects that we weren't even able to meet up with them, but we wanted to make that connection, bridge that that space and that time, and start creating a relationship so that we can make this program successful in the years to come as well.
Yeah, so into the field work, we were covering Region 2, which you can see on the map there. We'll cover some of the resources that we used this year in the field, some of the phenology that we found and useful things along with that. The Smut Fungus Challenge being our largest challenge this year, we'll go into more details on that, as well as the general nature of the field work that we conducted this year and some blips and challenges related to that.
All right, so doing our field work, this season it was just Allie and I, so it was 2 techs. We covered, Region 2 covers 5 states, though you can see from this picture on the slide, we mainly focused our efforts in Colorado and Wyoming, and 1 visit up to the Black Hills National Forest in South Dakota. This is a pilot year, so there were a lot of things to still be discovered. And again, grasses are hard. So there was a lot of work in getting familiar with our target species and getting out there in the field and starting to identify them.
Yeah, and so into some of the resources for our scouting and collection operations, SciNet points were incredibly useful. So we thank Marty Aitken for providing those to us. It's especially useful for knowing if there might be a multi-collection site, what the elevation might be, what the slope might be, as well as how close it might be to major roads, as all of those things are things we like to consider, especially if we're going to do a collection with volunteers or in a large group setting so that it's more safe for the group. So here's just an example of a multi-collection from a SciNet point. You can see all of those different colors represent a different species that's in that same location, which is super helpful for us to know if we wanted to make a multi-collection point. The next resource we had for scouting was our Forest Service partners. They were incredibly helpful, providing us with on-the-ground support as well as coordinate points, great email advice on which species we should be looking for and where they might be and how they might be looking at their stage of development at the time. So we really, really appreciate the Forest Service partners for our scouting operations. And finally, if we weren't really able to access either of those things, we would do some roadside botanizing. So this just means looking out the car window, seeing if there might be a huge field of Festuca arizonica, Festuca idahoensis out there, and then, yeah, just going out and collecting.
So one of the challenges that we ran into this year was the phenology. This year, 2023, was a high precipitation year across what seemed like the whole region to specifically Colorado and Wyoming. And we found, at least in Colorado, it was cool and wet for many months, and then all of a sudden it turned very hot and very dry, which gave us in late August and into September and even a little bit into October, a very narrow window between mature seed and seed that had already fallen. So that was a bit of a challenge this year. Plus, because of that high precipitation year, it was cool and wet, everything was kind of delayed. So we weren't able to get into the field until July 10th. That was our first collection date. And the picture here you see is out at Thunder Basin National Grasslands. And while we were collecting Nacella virigula, I looked over the hill and saw Allie collecting, and off in the distance there you can see a flare stack. So it was a really interesting place to collect from, especially for our first field visit.
Yeah, so into the smut fungus. This was a huge issue for us this year that we didn't expect at all. We believe because it was such a wet year for Colorado, this fungus really thrives. It thrives in wet conditions and can wait in root systems for a very long time. And some of the things that we've learned about it this year include that it mostly affects graminoids. And we found that provisional seed zones that tended to have less precipitation and were less humid had a larger outbreak of the smut, potentially because they were wetter, much wetter this year than previous years, as well as slope and aspect having an effect on this, including larger, steeper slopes with less of the fungus, hot and dry areas with less of the fungus, and then also that south-southwest facing slopes having less of the smut fungus present. But we still have a lot of questions about this, including, can it cross-contaminate across species? If we were to collect something that had fungus, is it safe to keep with collections that may not have fungus? Can we collect forbs when there's evidence of smut on the grasses? Can it spread onto our forb populations?
So moving into results, Allie and I covered 9 forests and 2 grasslands this season. We made 48 individual collections with 143 data sheets, and these data sheets include not only the information and the data for the collections that we made, but also in areas that that either were great to collect in, but the seed wasn't mature yet, or maybe the seed had already shattered. It also includes negative data sheets. So that means if we followed a SciNet point and we got to an area and we didn't see any evidence of the species that had been collected there before, we would make a negative collection data sheet showing that it was not worth going back to this area for future crews. And altogether, we covered about 10,000 miles, and in that time we only got 3 flat tires and 1 cracked windshield from these golf ball sized hail that we encountered out of the Black Hills when we were in South Dakota.
Yeah, so continuing on with those results, we collected 16 species this year, which was 12 grasses and 4 forbs. Some of these were opportunistic species, but almost all of them were target species for this year. We collected across 4 ecoregions on the Almernick Level 3 and across 5 provisional seed zones. And the real kicker is that we were able to collect 26.56 pounds of seed this year, which is huge. And we also want to point out the fact that these larger collections were a result of CONUPS events. So our volunteers were incredibly helpful to making sure we could bulk out the seed for this year.
And moving into some of the lessons that we learned this season, is as far as training goes, again, if we haven't already made it clear, grasses are hard. And so we feel that crews would really benefit from some additional training, specifically get some more time in the herbarium to look at these species, because even within a single species, you can have a lot of variation. We also discovered early on in the season, as we were bringing samples back to the office, that we had started to amass quite a collection of species to identify here back at the office, and we thought that for the following year, having some disposable samples for crews would be really helpful early on in the season when grasses haven't started to flower yet, and they're able to take these samples and actually pull them apart and really look at them and look at these specialized morphological features of grasses that you can't do with herbarium samples. We also believe that some training in being able to identify the target species at every cycle in their life. So just vegetative, also vegetative before it starts to flower, and also what it looks like after the seed has shattered. So that way when they're in the field, they can take a look at a population, be able to identify it. Of course, if there are morphological characteristics in which identify them as that species, without the flower, but then they can go out there and collect data so that they know in the following year, this is definitely a place that a crew should come and visit and make a collection. We'd also like to include training for lookalikes and input those into our SciNet points. And this is going to be really helpful, especially for species that should definitely not be in our collection, such as quackgrass, and being able to know specifically how do you tell the difference between Elamis repens and Pascopyrum smithii. And finally, we thought that maybe having a day of training of plant pathogens would be really helpful, because smut is not the only fungus that's out there. It's not the only pathogen that affects plants. And being able to come up with a protocol that crews can follow would be really helpful to make them more efficient in making decisions out in the field, whether they make a collection or they don't. And if they do, what are the protocols that they follow when they collect a plant that has evidence of some kind of pathogen.
Yeah, and probably the most important lesson that we learned this year was that the more engagement we had with the partners, the better it went off for us. So we just want to expand this for the following year, which includes us creating educational materials. So right now we're in the process of amassing a series of videos, guides, and different tools within this project's OneDrive so that next year's crew has more engagement with previous year's work. We also want to engage more with our CONIPS volunteers and CONIPS partners, as again, these collections were our largest and most successful, and we really feel that the educational opportunity that we can provide is essential to this project as well. We also would like to include range folk from the Forest Service more, as they have great on-the-ground experience about grasses and great tidbits on how to identify between species and distinguish between them as well. And they just are a great source of knowledge that we did not utilize as much as we could this year. And finally, just more outreach with all of our Forest Service partners. So just making sure to send those emails early in the year, seeing when they might be able to come out into the field with us, doing visits to the offices as well as to their forests. So yeah, we would love to do more of that as well.
So thank you very much. If you have any questions, you can reach out to Jessica Smith, who is the sort of the director of this program at CNHP. She's the main point of contact. And if you have any questions, any feedback, any insights for this project, would be greatly appreciated. And thank you so much.